Hello, everyone. In the previous lecture, we learned how T cells become activated through antigen presentation and how helper and cytotoxic T cells coordinate cellular immune responses. However, many pathogens spend at least part of their life cycle outside of cells. Viruses move between cells, bacteria may live in extracellular spaces, and many toxins circulate freely through the body. To defend against these threats, the immune system relies on B cells and antibodies. In this lecture, we'll examine how B cells become activated, how antibodies are produced, and the many ways antibodies help eliminate pathogens. By the end of this module, you should be able to explain how B cells become activated, describe the function of antibodies, and differentiate among the major antibody classes. One major difference between B cells and T cells is how they recognize antigens. T cells require antigens to be presented on MHC molecules. B cells do not. Instead, B cells use membrane-bound receptors called B cell receptors or BCRs that can bind directly to intact antigens. When a B cell encounters an antigen with a shape that matches its receptor, the antigen binds to the BCR and initiates the activation process. This direct recognition allows B cells to detect pathogens and toxins circulating throughout the body. Not all B cell activation occurs in exactly the same way. Most B cell responses are T cell dependent. In these responses, a B cell binds an antigen, processes it, and presents fragments to helper T cells. The helper T cell then provides activation signals that stimulate the B cell to divide and differentiate. T-dependent responses tend to produce stronger antibody responses, better memory formation, and longer lasting protection. Some antigens can activate B cells without T cell help. These are called T-independent responses. Although they can provide protection, they generally produce weaker immune responses and less immunological memory. For most of the immune responses we'll discuss, helper T cells play a critical role in supporting B cell activation. Once a B cell is activated, it undergoes clonal expansion. The activated B cell divides repeatedly, producing thousands of genetically identical daughter cells. Because all of these cells originated from the same parent cell, they recognize the same antigen. These daughter cells can follow two major pathways. Some become plasma cells, which are specialized antibody producing cells. Others become memory B cells, which remain in the body long after the infection has been cleared and provide long term protection. Antibodies are proteins. Uh, sorry, excuse me. Antibodies are proteins produced by plasma cells. Each antibody is highly specific and binds only to antigens that match its binding site. Once secreted, antibodies circulate throughout the bloodstream, lymphatic system, and other body fluids where they can encounter pathogens. Importantly, antibodies do not usually kill pathogens directly. Instead, they mark pathogens for destruction or interfere with their ability to infect the host cells. You can think of antibodies as highly specific targeting molecules that help direct other components of the immune system towards the correct targets. The immune system produces several different classes of antibodies, each with specialized functions. IgM, or immunoglobulin M, is usually the first antibody produced during an infection. It is particularly effective at activating complement. IgG is the most abundant antibody in circulation. It provides long-term protection and is often the dominant antibody produced after vaccination or repeated exposure. IgA is found primarily on mucosal surfaces such as the respiratory tract, digestive tract, and saliva. It helps prevent pathogens from entering the body. IgE is involved in defense against parasites and also plays a central role in allergic reactions. IgD is found primarily on the surface of B cells, where it functions as part of the B cell receptor complex. Together, these antibodies classes allow the immune system to protect many different parts of the body. One of the simplest and most important functions of antibodies is neutralization. Many pathogens must attach to host cells before they can cause infection. Similarly, many bacterial toxins must bind to specific receptors to exert their effects. Antibodies can bind to these pathogens or toxins and physically block their ability to attach to the host cell. By preventing attachment, antibodies neutralize the threat before infection or damage can occur. This mechanism is especially important in protection against many viruses. Another major antibody function is opsonization. The term opsonization means to prepare for eating. When antibodies coat the surface of a pathogen, immune cells such as macrophages and neutrophils can recognize the antibody-coated microbe more easily. Special receptors on the phagocytic cells bind to antibodies and help pull the pathogen into the cell. As a result, opsonized pathogens are engulfed and destroyed much more efficiently than uncoated pathogens. Antibodies can also cause agglutination. Because each antibody has multiple antigen binding sites, a single antibody can bind more than one pathogen at a time. As antibodies bind multiple microbes, they can link pathogens together into large clumps. These clumps are easier for phagocytes to capture and remove from circulation. Agglutination also helps limit the spread of pathogens throughout the body. Antibodies can activate a powerful collection of proteins known as the complement system. When antibodies bind to the surface of a pathogen, they can trigger a cascade of complement proteins. Complement activation can enhance inflammation, attract immune cells to the site of infection, and increase phagocytosis, as well as, in some cases, directly damage microbial membranes. Because complement amplifies immune responses, it is an important partner of the adaptive immune system. Another mechanism involves cooperation between antibodies and immune cells. In antibody-dependent cellular cytotoxicity, or ADCC, antibodies bind to infected cells. Natural killer cells, often called NK cells, recognize these antibodies through specialized receptors. The NK cell then releases cytotoxic molecules that kill the antibody-coated target cell. In this way, antibodies help direct immune cells toward infected or abnormal cells that need to be eliminated. Not all activated B cells become plasma cells. Some differentiate into memory B cells. Memory B cells persist long after an infection has been cleared. They retain information about the specific antigen that triggered their activation. And if the same pathogen enters the body again, memory B cells can rapidly expand and produce large quantities of antibodies. This faster and stronger secondary response often prevents illness entirely. Memory B cells are one of the major reasons people typically develop immunity after infection or vaccination. Let's review the major concepts from this lecture. B cells recognize antigens directly using B cell receptors and can be activated with or without helper T cell assistance. Activated B cells undergo clonal expansion and differentiate into plasma cells and memory B cells. Plasma cells produce antibodies, which bind specific antigens and help eliminate pathogens. Antibodies protect the body through several mechanisms, including neutralization, opsonization, agglutination, complement activation, and antibody-dependent cellular cytotoxicity. Finally, memory B cells provide long-term protection and allow the immune system to respond more rapidly during future exposures. In the next lecture, we'll see how immunological memory can be generated intentionally through vaccination and explore the immune response to mRNA vaccines using COVID-19 as a case study. With that, thank you so much for attending this lecture, and I'll see you at the next one.